Welcome. Here we are again, talking about kingdom Protista. Last time we discussed the subkingdom Protozoa, which are those heterotrophic unicellular animal-like things, for lack of a better way to describe them. And we went over the taxonomy of the subkingdom. We have these four phyla. Remember, we use the word phylum here because they're more animal-like. And then we get to the subkingdom Algae, and that's what we're going to do today. As far as the differences between the two subkingdoms, and like I mentioned before, sometimes the kingdom Protista is described as a garbage kingdom because there are seemingly unrelated things dumped into the same kingdom, and often taxonomists joke that, well, if we don't know where it goes, just dump it in the Protista. There are some things that link the two subkingdoms together. One is life cycles, similarities in life cycles that we see. As far as differences go, there are lots of differences. And the primary one that we're going to look at is that the subkingdom Algae is photosynthetic. They're autotrophic. So let's think about that for a second. And so we might say, okay, well, one thing that links all these together is that they're autotrophic. Specifically, they're photosynthetic, okay? And they are, because of that, often referred to as being plant-like, but the thing is, some of them are unicellular, and they don't, when they're unicellular and flagellate and they're swimming around, they don't really look like plants. The only thing plant-like about them, perhaps, is that they're photosynthetic. The way that we split these up is somewhat artificial. Some of it has to do, at least with these first three, with combinations of pigments that are produced, the greens, the reds, and the browns. What makes them red is a pigment called phycobilin, and then different combinations of other pigments are what give them their distinctive colors. Okay, so there's a lot to discuss about each of these groups. I will tell you that the first three are primarily multicellular. I mean, you see a lot of multicellularity here. These last three groups are unicellular exclusively. And so what do we have? They all end in phyta. Phyta is phycologists, or the people that study algae. So really, you're just worrying about the prefixes chloro, rhodo, pheo, chryso, pyro, and eugleno. And I think I mentioned this in the last video. This is one of many, many different ways that you could classify the algae. I'm a lumper. I'm lumping for the sake of time, really, more than anything. If you look at the newest taxonomy, you'll see they take the Chrysophyta and they split it up into two or three different groups, divisions. If you want to take the time to really learn the taxonomy of algae in its most modern form, then go for it. Okay. It's not to say that one is correct over another necessarily. It's a lot of it is a matter of opinion. Okay. So we're going to start with the greens, the chlorophyta. And this is exciting, y'all, because guess what we get to do now? We get to, this is going to be our introduction into the idea of a life cycle, okay? And the idea of a life cycle is you follow the organism through one generation. It's kind of like doing a Mendelian genetics problem, except you're looking at morphology and habits and ecology and stuff like that in terms of their life cycles. So the chlorophyta, the green algae, are by far the most diverse of any algae division, meaning we find them in freshwater and marine habitats. Darn it. I hate when that happens. Oh, I've done it now. Shoot. Sorry, team. There we go. Okay. This thing is really sensitive. There. Okay. So we find them in both freshwater and marine habitats. And the thing that makes them, one of the things that makes them the most diverse, honestly, is their different life forms. And so what is a life form? It just literally means morphologically, what does the thing look like? Well, now look at all these different life forms we have. We have unicellular. We've seen that before in the bacteria, right? Colonial, we talked about colonial a little bit in bacteria as well. Filamentous, and then we haven't really talked about phallic. So let's just take a second to think about this. Unicellular is pretty straightforward. There's one cell, right? Typically, the unicellular ones have flagella, and they literally swim around in the water, but they're photosynthetic, see? So that's the thing to think about. You know, if they weren't photosynthetic, what would they be? Well, they would be flagellates, but they are photosynthetic. They're green, okay? We're going to look at examples of these, each of these life forms. Colonial is where you have a colony of individuals, still photosynthetic, mind you, living together like this. And their little flagella poke out of the colony. The colony that is what they're embedded in is sort of usually just this like gelatinous goo. I know that sounds funny, but that's what it is. It's a gelatinous matrix. So here's colonial. Okay. Filamentous, now we talked about filamentous already with the cyanobacteria, but guess what? Now it's a little bit different. I'm going to draw some nuclei in here. And now what we're talking about is mitosis in one plane. Every time mitosis occurs, it can only occur in this one plane. What that does is it makes the organism can only get longer. It can't get any taller or thicker. Okay. Before we weren't talking about mitosis with the bacteria because they don't do mitosis. They don't have a nucleus. Okay. And then phallic, so this is filamentous. And then the last one is phallic. And here's phallic. See if you can figure this out. And again, these are all photosynthetic organisms, so they would be green as well. Okay. Phallic mitosis occurs in how many planes? In 2 planes, like this. So the organism can do mitosis in 2 planes, and so it makes this flat sheet of cells. It can get longer and taller, but it can't get any thicker. Okay, and so we'll see examples of all of these. We're going to go through and we'll look at an example of a unicellular green algae and a colonial and a filamentous and a phallic. Okay, they're important as producers of oxygen. Guess what, y'all? The algae, not just green algae, but the algae as a subkingdom, produce 80% of the oxygen that's liberated on Earth. That means, you know, you hear a lot about plants and oxygen, but the reality of it is that the algae are making most of the oxygen. How could this be? Well, most of the Earth is covered with water, and water has depth. It's 3-dimensional. And so, you know, there's a great deal of photosynthetic activity that can occur in the depths of ocean, okay, and freshwater. Okay, so let's look at our first little green algae. And it's called Chlamydomonas. Let me clear all the drawings there. Okay. Check out what you have here with Chlamydomonas. It is and just so you know, this is a genus name. And generally, I won't hold you to the genus name. You know, we're studying this stuff on the level of kingdom, subkingdom, and division. And so what are we looking at here? Well, here's a great big chloroplast, right? And here's a pair of flagella. There's one and another. And that's Chlamydomonas. It's a unicellular chlorophyton or green algae. What do these things do? They swim around in the water and they look for the light. And when they find the light, they're able to make sugar. So they're motile. Okay. Chlamydomonas, it turns out, is going to be our first life cycle. And the thing about Chlamydomonas is it's an excellent first life cycle because it's literally the simplest life cycle you could ever really have in an organism, and we'll see why that is. Okay. Whenever we're talking about life cycles, we really have to think about sexual and asexual. Okay. And asexually, this organism, I think I'm gonna go for a whiteboard here for just a second. Just because it'll be easier for me to get where I need to be. Oh, look at that, genetics. Ooh, goodness. Okay, what are we doing? Here's Chlamydomonas. Life cycle. And it's our first one, and I'm so excited to start life cycles. Okay, here we go. Asexual reproduction in this organism, there's an individual. We'll give him a little happy face, a little smiley face there. Mitosis, right? Now, a curious thing about this organism, we'll say times up to 3. And so think about it now. If this single-celled organism drops its flagella and does mitosis, you're going to get 2 cells. And then if each of those 2 cells does mitosis, you get 4. If each of those 4 does mitosis, you get 8. This should actually be 4. And then if each of those 8 cells does it, you get 16. You get a doubling of the number of cells. So what we then have is this little shell with all these little baby chlamydomonases, chlamydomoni maybe you would say. I'll just draw a few more. We're going to say up to 16 new daughter cells. And because this is mitosis, you know, these things sort of hatch out and off they go, dun, 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 swimming away. And then you're back to here. If this is mitosis, what do you know about mitosis? One thing about mitosis is it doesn't change the ploidy. Turns out that Chlamydomonas is a haploid organism. And so all these little individuals in here would be one end. They have one set of chromosomes. Now, humans are diploid organisms, and so this might not make a lot of sense, but it just turns out that some organisms can be haploid in their adult phase of life, okay? This is an adult individual of this unicellular photosynthetic organism. So here's the thing, y'all. As long as the environment is good, Chlamydomonas is pretty happy to make clones of itself. Imagine if you could just make 16 clones of yourself at will. Well, that would be great, wouldn't it? I could be here teaching, I could be in the kitchen making lunch, and I could be riding my bike all at the same time. Well, Chlamydomonas can do that. It can clone itself through mitosis since it's a unicellular organism. Pretty convenient. As long as the environment is good, generally Chlamydomonas is pretty happy to just do asexual reproduction. It's when the environment starts to peter out on Chlamydomonas, and what does that mean if you live in water, right? This organism lives in water. It means maybe the water's starting to evaporate, or maybe there's some other tonicity issue with the water, right? This is typically what will trigger sexual reproduction in this organism. So let's think about sexual reproduction. And remember now, from your study of genetics, in order for it to be sexual reproduction, two things have to happen. One of the things that has to happen is fusion, and the other thing that has to happen is meiosis. Both of these have to occur for it to technically be sexual reproduction. So we're going to, in this introduction to our life cycle, we're going to see both of these things happen. Now, let's just say here's a little chlamydomonas individual, and it's 1N, okay? So the clever thing about chlamydomonas is because it's haploid and because it's unicellular, it can just, at any given point in its life, decide to be a gamete, right? So this little guy's going to say, I'm going to be a gamete, exclamation point, right? It can do that because it's already unicellular and it's already haploid. And now along comes another chlamydomonas. It's also happy, and this one says, me too, okay? So these organisms can just be gametes at any point because they're haploid and unicellular. Very cool, huh? Okay. Well, are there boys and girl? Are there boy chlamydomonas and girl chlamydomonas? No, there's not. What we have are mating strains. You can't look at one chlamydomonas and tell it from the other. They look the same. We typically indicate the mating strains as being plus and minus. We'll talk about types of gametes in a second. Let's make it through this life cycle, okay? So what do gametes do? If these decide to be gametes, by definition, a gamete is a fusion cell. So fusion, because these organisms are haploid dominant, fusion occurs first, okay? And then we get this cell, and this is the first cell of the new generation. What's its ploidy? Well, 1 + 1 is 2. This is diploid, and this is called the zygote. The zygote is the first cell of the next generation, okay? Now, we got to get back to haploid, right? Because the adult chlamydomonas, your average everyday chlamydomonas swimming around in the pond, is a haploid organism. How do we get to haploid? Well, it's pretty simple, right? We do meiosis. So this one zygote, this one cell does meiosis. Remember, when meiosis occurs, we get 4 little flagellated swimmers, okay? And these individuals, it turns out, 2 of them are going to be pluses and 2 of them are going to be minuses in terms of mating strain. And we call these little boogers zoospores. And a zoospore is just a product of meiosis. Zoo because it swims, right? And then nothing really has to happen to get back to here. These things just swim away and they grow up into chlamydomonas, okay? Now, there's one little thing that happens between these guys here and fusion, and I should have drawn it in, and it's called clumping. So I'm going to draw clumping right here. Clumping is where they come together and they drop their little flagella. They put their little butts together. I'm going to make them kind of frowning because they're a little embarrassed. So they drop their flagella and they put their little butts together and they start rubbing their little butts together before they fuse. And this is called clumping. Now, crazy thing about clumping is all it takes is for one pair of chlamydomonae, and you can imagine these are microscopic organisms. There can be literally hundreds of thousands or millions in a jar, if you think about it. They're very tiny. All it takes is for one pair to start clumping, and it literally creates this sort of orgy of chlamydomonas clumping and fusing. And so this one pair, pretty soon some others will come and join in, and then there's this big ball of chlamydomonae, and they're all looking for mating partners to clump and fuse with. Okay, so that's an interesting little thing to see. Often when we order these organisms, and I apologize because if we were in an actual lab, we'd get to look at these under the scope, and I'm sad we don't get to do that. But often the stress of transport causes them to engage in sexual reproduction. And so often when we get them, we'll see them and they'll be clumping, or there'll be a lot of zygotes floating around in the test tube or the Petri plate. Okay, so why wait until the environment's going bad? Well, why not do it then? Because what you're accomplishing here is a genetic recombination, right? You're getting new genotypes here in the zoospores, and so maybe one of these genotypes will be better able to survive in the petering out environment. So this is the first life cycle. You can see we've completed the life cycle. We've done both fusion and meiosis. We had to do fusion first because these organisms are haploid dominant. We're going to see this a lot. Okay, this is very common in living things. This life cycle has a name, and it's called the zygotic life cycle. And it's called the zygotic life cycle because if you look, you will see the only diploid cell, the only cell that has two sets of chromosomes in this entire life cycle is the zygote. Everything else is haploid. Okay, so things that exhibit the zygotic life cycle, the only diploid cell is the zygote. As soon as the zygote is formed, the next thing that happens is meiosis, and you're back to haploid. The zoospores are haploid. Okay, so there you have it. Now, we're going to spend just a second talking about gamete types, and hopefully this is a good little first life cycle for you, because they get more complicated. So I'm going to tell you, if this doesn't make sense to you at this point, rewind the tape and watch it again, because this is the simplest a life cycle could possibly be, and they're going to get more complex. But the more life cycles we look at, actually, they're all basically the same processes of fusion and meiosis. So they tend to get easier over time, even though they get more complicated. Check this out. There are different kinds of gametes. Isogamy. Isogamy is what we just saw, where you have two gametes and they're both motile, meaning they can both swim. They both have flagella, or they're flagellae, or they have some means of motility, and same size. Iso means the same, isogamy. Okay. There's also something called anisogamy. I don't think we actually do a life cycle with anisogamy, but it's like this. Okay. So they're still both motile. Of course, there's two. They're still both motile, but one big and one little. We'll just say one's bigger. Okay. These are mating strains. And then the last possibility is called oogamy. Oo means what? There's a little umlaut. That's a German thing over the O. Oo, ovum, ovary. What does it mean? It means egg, right? Egg gamete. And so in oogamy, you have a large non-motile gamete and a small motile one. What do we call these? Egg and sperm. So what kind of gametes do humans have? We exhibit oogamy. We have an egg and a sperm, right? And the one that we saw earlier with Clammy was isogamy. Okay. So this is just a little, a little additional information that you will need. Some algae actually exhibit oogamy. We'll see this in just a second. Okay, let's continue on then. Now we get to talk about our colonial life form. And our colonial life form example is going to be called Volvox. And it's curious what you're looking at here. Volvox is this very bizarre organism. We may have to watch a little video of it just for fun. What you have with Volvox is a colony of Chlamydomonas-like organisms. And so what you have is this gelatinous matrix, and embedded in the gelatinous matrix are all these little Chlamydomonases, Chlamydomoni-like organisms. I'm just going to draw a bunch in here for a second. All of their little flagella poke out like this to the outside of the colony. Okay, and it's so interesting. There's always 256 individuals in a Chlamydomonas colony. Isn't that strange? There's always 256. What's the relevance of 256? Well, how is the colony made? You take one Chlamydomonas-like organism, and that one Chlamydomonas-like organism begins doing mitosis, right? And in the process of mitosis, how many mitotic divisions in a row to make 256? Well, let's figure it out. 1 makes 2, there's 1. 2 makes 4, 8, 16, 32, 64, 128, 256. So 1, 2, 3, 4, 5, 6, 7, 8 mitotic divisions in a row of starting with 1 cell would give you 256. That's how the colony forms, right? And once you have this colony, what can then happen is cells within this mother colony can begin to do the same process of mitosis to make what you see here, which are daughter colonies. And it's really bizarre that these daughter colonies form inside the mother colony. And then eventually the mother colony just sort of splits open and out come these little babies. And then they go on to become adult Volvox colonies. Very strange. Okay. What does this thing do? Well, it can rhythmically flap its flagella. These cells can communicate with one another, and this thing can just roll through the water. It's really very cool to see. There used to be the live stream Volvox channel where you just put it on and it was just, all it was just live stream Volvox. Okay. Volvox, in terms of its life cycle, exhibits oogamy. And so what you have is some colonies will let this one, some of the cells will sort of round up and lose their flagella. And so now this would be sexual reproduction. And they make eggs, right? And so another colony over here might generate through mitosis some sperm that it releases into the water. And this egg comes and swims over here and finds or the sperm rather swims over and finds the egg, and then we have a zygote in the colony, right? So I won't hold you to the details of the Volvox life cycle, just know that it's oogamy. And so we have some colonies making eggs and some colonies making sperm. In fact, the colonies can make both. So this one colony could make eggs and sperm. And now, would it make sense for the sperm to fertilize its own egg? Not really, right? The whole purpose of sexual reproduction is genetic variability. And so you don't want to have sex with yourself, is basically what that would be. So there's a strategy that's employed to prevent this from happening. And the strategy is, it's a temporal separation. That means time. It's a temporal separation of time between when the eggs are ready and when the sperm are ready. Typically, the sperm will develop first and be released into the water, and off they go to look for another colony. And then only later do the eggs of that colony develop. And then this allows the sperm of some other colony, theoretically, to come across the water and find them. Okay, so very cool. Let's just take a second to look at a short video. And we don't indulge ourselves very often. By doing this. So let's do it. Let's look at a video. Let's get out of this for a second. Oops, shoot. no, that's not what I meant to do. Okay. Let's just check out a Volvox video just for fun. Here it looks like a pretty good one. This is the first one that pops up. Now this one has, oh, here's one of Volvox releasing daughter colonies. That might be cool. But let's watch this one first. Very dramatic music. Okay, that was pretty cool. I liked that.
The ultimate
Wow, look at all those, y'all.
alien evolution is Volvox, whose beautiful colonies are large enough to be visible with the naked eye. The eye of the needle gives a sense of scale. Each colony is a sphere consisting of hundreds of tiny somatic cells and a few much larger reproductive cells called gonidia. Most gonidia in this sample have already started forming embryos, so individuals may contain several generations of colonies. Starting.
Okay, there you have it. Everything you ever wanted to know about Chlamydomonas, about Volvox. It's right there on the internet. What about a filamentous green algae? So we saw unicellular Chlamydomonas. We saw colonial Volvox. Now there's one called Spirogyra, which is a filamentous form. And in Spirogyra, the name comes from the spiral chloroplast, which is very cool. You can see this is a chloroplast here, and it's spiraled. It's like almost like a DNA helical shape inside the cell. And you can see it's filamentous, right? There's the cell wall and another cell wall. So this marks one cell right here. Okay. Spirogyra is beautiful to look at under the microscope, and if you want to take a second, you can you can always go online and look at some images of these. And Spirogyra is common in freshwater, stagnant freshwater, but not really super poor water quality. So, for example, abandoned swimming pools or ponds, if you see the green slimy stuff, sometimes that'll be Spirogyra if you pop it under the scope. Okay. Spirogyra does this weird thing called conjugation for its sexual reproduction. Its gametes don't quite fit into those groups that we gave. It's a little bit strange. And again, we won't hold you to the life cycle for Spirogyra. We might check out another view of it though. Just because you're only seeing a couple of cells there, it's not really doing it justice. Oops, let's just take a look at some more images of it. Now there's also a band called Spirogyra. They spell it with They spell it with a Y, actually. They stole their name from an algae. That's a nice picture. Look at this one. And see, you can see how pretty it is with its spiral chloroplast. Very distinctive, hard to misidentify with anything else. And then also, let's look at conjugation. Let's look at this picture. And again, you're not specifically responsible for the life cycle of Spirogyra, but I think it's pretty cool to see conjugation. And that's what's happening here. So what you have is this situation where these little two filaments will come sort of line up beside each other. And then they'll form the cells of one filament and another will form this little tube, similar to a conjugation tube that we saw in the bacteria. And what then happens is the nucleus from one of these filaments migrates through the tube and fuses with the other. So these things are haploid, right? And so you could imagine all the nuclei migrating and fusing and migrating and fusing and migrating and fusing. What are we forming in each of these cells? Zygote, 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 right? It doesn't quite fit with other types of gametes because the gametes are literally, they don't have flagella but they're kind of motile in that they're migrating. Okay. So that's what you're looking at here. All right. We're going to do, let me check the time here. Yeah, we're going to do one more life cycle today. It's going to be interesting. And while let's see. So here's ulva. Now we have a phthalate green algae, and you've probably seen ulva, honestly. You've probably seen it at the beach. You might have been walking in the shallows of the water and some green slimy thing crept up on your leg and it probably freaked you out a little bit. That's ulva, right? Ulva is commonly called sea lettuce. And it's often found on pilings of piers, you know, what holds the pier up at the beach. It's typically anchored down onto something. And so it consists of this sort of blade-like structure, or it's just commonly called a blade. And then this little thing that holds it down is called the holdfast, kind of like the roots, if you will. Of course, they're not absorbing anything. Why don't they need to absorb anything? Well, because this organism is bathed in ocean water, and ocean water has all of the nutrients that anything might need. Okay, so there's an interesting story about ulva. For a really long time, oh, let me point out, it's phthalate, right? So it's only a single cell layer thick. You can see how thin it is. You can look right through it, right? It's only a single cell layer thick, but it's kind of plant-like in a way. You know, it has a blade, kind of like a single leaf. And for a long, long time, scientists could not quite figure out the life cycle of this organism. They would collect it and grow it in aquariums and try to get it to reproduce in an aquarium, you know, and go from an adult ulva to another adult in the aquarium. And they never could quite figure it out. Okay. It was assumed that it exhibited the zygotic life cycle. It was actually a graduate student at a university that was studying algae that said, well, has anyone ever bothered to count the chromosomes in the cells of the adults? And everyone was like, no, I don't think we've done that. And his point was, how do we know that the adults are all haploid, right? Maybe some adults could be diploid. Maybe they could have two sets of chromosomes. So that was what he did, and this is what he discovered. And so we're going to do this life cycle. It's one of the more complicated life cycles that we'll actually cover in this class. Okay. Let me get rid of these drawings. And get excited because this is a fun life cycle, let me tell you. Woo! Sex lives of algae. Something else, y'all. Okay. So let's see. Let's go back to here, to our sketch pad. And here's how the life cycle starts out. Here is one ova, and we're going to let this be a plus strain. And here is another ova, and we're going to let this be a minus strain. Now, these are adult individuals of this organism. They're phallic, right? They're multicellular. So this is the most complicated life form that we've seen thus far in the class, right? At least since we started doing our kingdom surveys. I mean, think about the bacteria. They were very simple. At best, filamentous in the cyanobacteria. Now we have this thing that's sort of multicellular, right? And these individuals we're going to define as being the gametophytes, or the gametophyte generation. And you could bet that the gametophyte generation is haploid. It's 1N. So every cell in these individuals is haploid. It's 1N. And you could imagine what the gametophyte generation is going to do. It's going to make the gametes, right? Now, if you have this organism and it needs to make a unicellular gamete, how can it do that? It's already haploid. And so what can literally happen is mitosis. And along the edge of the blade right here, mitosis occurs. And what is produced is a little flagellated swimmer, right? And then the same thing is going to happen here. We'll get a little flagellated swimmer here. And what are we going to call these? If I tell you they're about to fuse together, we're going to call them gametes, right? So these are the gametes. They're haploid, right? Remember, mitosis doesn't change the ploidy. And so if this haploid gametophyte does mitosis, it's going to make a haploid cell. So there's the two gametes. By definition, what do gametes do? They fuse. And what do you get? You get a zygote, which is diploid. Now, what the scientists that were studying this were assuming was that this zygote does mitosis, just like it did in Chlamydomonas. If you got rid of this part, with the adults and you just looked at this, you'd think, well, these individuals are, it's just the Chlamydomonas life cycle right here, okay? Well, what they assumed was meiosis occurred to restore the haploid condition and then some mitosis to make the new adult generation, but that's not what happened. Instead, the zygote doesn't do meiosis right away. It does mitosis times many. And what do you get? You get an adult like this. And I've drawn it bigger, but it's no different morphologically from the gametophyte generation. We're going to call this diploid generation the sporophyte generation. It's 2N, okay? From the zygote, we did mitosis times many. Mitosis doesn't change the ploidy. One cell to many cells, the ploidy stays the same, okay? This was where they were going wrong because morphologically, you can't tell the difference between a sporophyte or a plus strain gametophyte or a minus strain gametophyte. So when they would go out and collect random ova from the ocean and put it in an aquarium, they may not have had everything they needed to complete the life cycle. That's the point, okay? It was the graduate student that was like, count the chromosomes in the cells. And when he got to doing that, he realized that some of these adults had twice as many chromosomes in their cells as others did, okay? Well, now how do we get back to haploid? Well, you know the answer to that, right? You do meiosis. And so along the blade here, meiosis occurs as a process, and you get 4 little flagellated swimmers. Now we've restored, now these are the zoospores, same word as we used before, and they're 1N. And then now, think about this. How do we go from a single cell haploid to this multicellular gametophyte generation? Well, mitosis times many. And we're back where we started. Okay. So there are lots of things I could ask you about this life cycle. I could ask you, what's the name of this cell? It's the zygote, or this cell, it's the gamete. I can also ask you about the ploidy, and the ploidy becomes very, very important now. What's the ploidy of the gametophyte generation? Gametophyte generation is always haploid. What's the ploidy of the sporophyte? The sporophyte is always diploid. The gametophyte makes the gametes. The sporophyte makes the spores. You could think of it that way. Okay? And we're going to draw this out in a simplified fashion in a second, and it'll hopefully by then be clear. Okay? So the other thing I can ask you is the processes involved. What's the process between here and here? Fusion, right? 2 things becoming 1 thing. When the ploidy changes from 2N to 1N, you know it was meiosis. When the ploidy changes from 1N to 2N, you know it was fusion. Both those processes have to occur. And then we also have mitosis in this life cycle. So this is a trippy little life cycle here. It's a lot going on, okay? What do we call this life cycle? Alternation of generations. Alternation of generations, the alternation between the gametophyte and the sporophyte generation. That's what we're talking about here. Now, it just turns out that we're going to see alternation of generations occur in every plant life cycle that we do later, but what we're going to see is that in plants, you can tell the difference between the sporophyte and the gametophyte. They don't look the same morphologically. So that's what makes ulva tricky to the, at first glance, is that you can't really tell a gametophyte and a sporophyte generation apart. So alternation between the gametophyte and the sporophyte. Hopefully that made sense. I would really encourage you to draw these life cycles out like I have. And, it's nice if you can draw them out and label them, then you really know the life cycle, because you're going to be tested on this. Okay. What if I did this? Check this out. Look at this. This should make sense, right? This is just super simplified. I'm leaving lots of parts out, but this should make sense, right? Gametophyte to sporophyte through fusion, sporophyte back to gametophyte through meiosis, fusion, meiosis, fusion, meiosis. Every time you go around, you're in the next generation of the organism, okay? Good stuff, y'all. One last thing I might point out. We've seen 2 life cycles so far. We've seen the zygotic, and we've seen alternation of generations. What about us? What kind of life cycle do we have? Do we have either of these? Are we the zygotic life cycle? In the zygotic life cycle, the only diploid cell is the zygote. Well, we're not that, because all of our cells are diploid, except the gametes, right? We have sort of just the opposite life cycle of the zygotic life cycle. In the zygotic life cycle, the only diploid cell is the zygote. In us, the only haploid cells are the gametes. And so we're not going to get to do the kind of life cycle that we have, because we're not going to study zoology, and no other organism except animals has what we call the meiotic life cycle. And in the meiotic life cycle, the only haploid cells are the gametes. Everything else is diploid. So we're going to see a lot more of zygotic, and we're going to see a lot more of alternation of generations. We won't get to see this one. Sorry. But I just mentioned it to you because you may be wondering, well, what about us? How do we fit into this, right? We don't have gametophyte and sporophyte generations. We are sporophytes that directly generate gametes. We eliminate the gametophyte. Okay. Awesome, y'all. Well, we're going to stop there. The good news is those are the only life cycles for the algae, and the only ones that I'll hold you to really, and for the details, Chlamydomonas and Ulva. The rest of these groups, for example, next would be the browns. We're just going to talk fun about them and, you know, see how they're interesting or ultimately relevant to us. Okay. All right, signing off. I hope you have a great day. Oh, there I am, big on the screen. Hi. Take care. I'll see you next time.